Hello, welcome back. So this little mini lesson is just going to introduce some of the language and terminology of chapter 11. I'm going to try to hit on some of the meanings of the words that we're going to be using quite a bit to set you up for applying that and being able to solve some of these genetic problems. Okay, so we've seen the word allele before. It was introduced back in the meiosis chapter 10 and with your lab activities most likely. So here on this slide, we're going to talk about the definition of the word allele and introducing the word called loci, or locus is singular for the term loci. So again, when we talk about an allele, let's say we have a gene. A gene for, we'll take a look at this example here. Fur color, maybe we're talking about a cat or a dog or a bear or whatever. So here's the gene for fur color. It is located on this specific location of this chromosome. Let's say this is chromosome number 1. And these are homologous chromosomes. You should be having some familiarity with the word homologous, because remember, homologous chromosomes are the partners that lined up in prophase and metaphase 1 of meiosis and was separated in anaphase 1 of meiosis. So on chromosome 1 for cats, we'll say cat fur color. That's the gene is fur color. There are two options. You could have black fur or brown fur. Those options are what we call the allele. So it's the variation of that particular gene. So the gene is fur color, the alleles are black and brown. At a different location on chromosome 1, let's say there is a gene that codes for fur length. So the gene is fur length, the alleles would be long fur or short fur, okay? So we are taking a look at different genes. We have fur color at that particular location on the chromosome, and we have fur length at a different location on the chromosome. And then of those two genes, there's a couple different versions for each gene. So fur color, you could have black or brown. For fur length, you could have long or short. And we don't know in this particular picture which is dominant and which is recessive. We just know that those are two options. We're not given that further information. Okay, so the word loci is basically just referring to the location on the chromosome of that particular gene. It's kind of like its chromosomal address, if you will. So for fur color, the loci or the locus of that black or brown allele is at the kind of the upper end of chromosome number 1. It's going to have a particular location. We're not going to get into the super details of naming locations, but just know that a locus is the physical location of the gene. The allele is the different version of that particular gene. Okay, so looking back at some of these other chromosomes, if we look over here, this might be chromosome number 1, and this could be chromosome number 3. These are non-homologous, so they're not going to have the same genes on them. They're identifying the little yellow strips as loci, because that's just the locations of the genes, but they're non-homologous. They are not the same gene. So this gene is not the same thing as this gene. And the reason why we know that is because they're on two different chromosomes. Chromosome number 1 carries completely different genetic information than chromosome number 3. And this gene is not the same thing as this gene, right? So if we wanted to do not the same thing, okay? All right, so then it looks like in the second picture, kind of doing this might be number 1 from mom and number 1 from dad. So now we're back to homologous pairs. Homologous pairs. And so here we have a pair of alleles. Here we would have a pair of alleles. So these would be the partner genes with their alleles. And they're saying this one is not the same thing as this one in this little diagram here, which kind of makes sense because they're at different loci. All right, so then when we take a look at what I have down here in this bottom left, this picture is showing you the product of meiosis, right? So you just went through chapter 10. The end result of meiosis is 4 daughter cells with half the number of chromosomes. So in this case, if we had the diploid number was 8, which means you had 8 chromosomes, 2 pairs of 4. And the haploid number would be half of that, which would be 4, right? So in this particular diagram, we have a haploid sperm fusing with a haploid egg, and you can see they each only have 4 chromosomes, 4 individual chromosomes. They're not replicated, they're not in the homologous pairs, because at this point they're haploid, they only have 1 of each. When they fuse together, we are creating a diploid zygote, which now has 2 copies of each gene. Now they are in their homologs, right? So here's a homologous pair, here's a homologous pair, homologous pair, and homologous pair. It is on those homologous pairs where they will have the genes at the same loci, and they could have different alleles. They could be the same or they could be different. And so that's what we're going to be talking about a little bit later when we get into describing what we call genotypes and phenotypes. Okay, so this slide, I know I've written crazy numbers all over it. It is just to help you understand the definition of allele, which are different versions of the genes, and loci, which is the physical location of the gene on. Okay, so in this slide, you might be seeing in your reading what's called the law of segregation or the rule of segregation. And this is something that happens in meiosis. And so you have seen this before. We're just now calling it something specific. It may have even been mentioned in chapter 10. But the law of segregation means in the process of making your gametes, going from your diploid parent cell to your haploid daughter cells, you have your homologous chromosomes, right? So here's your homologous pair. In this particular case, because we're in metaphase 1, they're lined up, they're replicated, so they have their sister chromatids still attached. The parental homologs are next to each other. Those alleles, so in this case, they're showing the T gene, we'll say taco tongue, right? Taco tongue. In this particular parent cell, they have a dominant gene, that's written by the capital T, and the recessive gene is allele, sorry. The dominant allele is by the capital T. The recessive allele is designated with a lowercase t. So they have 1 of each. So this parent is what we call heterozygous, which we'll see on the next slide. The law of segregation means in the process of meiosis, those homologs, one's going to have the dominant and one's going to have the recessive, they will segregate, which is another word for separate. They are going to physically separate and end up in different daughter cells. So you already know this based on your understanding of meiosis. We are just calling it something new. So in this case, our dominant alleles have been separated from our recessive alleles. That's all it is. The law of segregation is just a fancy way to say the alleles are going to be dispersed. And so we see that here. In this case, a true breeding tall, when it segregates its alleles, all of the daughter cells are going to be dominant. In this case, when it segregates the alleles, all of the daughter cells are going to be recessive because that's all they have. If those two were to mate, right? So our parental generation gets our F1 generation, which is our hybrid. When those alleles segregate, they are going to split. Half of the daughter cells will be dominant and half of the daughter cells will be recessive. Okay, and if we cross-fertilize those, okay, this is where we get that 3-to-1 ratio, which again, we'll see when we get into the mini lesson on Punnett squares. So law of segregation is just the splitting of the alleles from the homologous chromosomes. They don't stay together. That's the whole point of meiosis, is to make the haploid gametes. So you take your sets of chromosomes and you break them apart into individual chromosomes, so 2n to 1n. Okay, and then lastly, these terms. This vocabulary is going to be so important when you're working on your genetics problems and trying to figure out and solve your genetics problems. So when we are setting up our solving of inheritance problems, we typically assign the genes and the alleles letters instead of numbers. Typically, and we always typically, well, I don't say always typically, that doesn't make any sense. It is convention, if you will, to give the capital letter to be the dominant allele, and the lowercase letter is going to be the recessive. Okay, that's just kind of how we do it, the rules, if you will, if you want to follow the rules. When you are assigning letters, you want to make sure you just use one letter per gene, not one letter per allele. So for example, if we're saying tall and short, it would be big T, big T, big T, and little t, little t. Those are the ways that we would express the true breeding tall, the true breeding short, and the hybrid. We wouldn't go tall, 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 short, and short, short. We wouldn't mix up the letters. That's a big no-no, because if you do that, you get confused because we're going to be dealing with lots of letters. So if you keep it, yes, with one letter per characteristic or one letter per gene, it's a lot easier to keep track of these things. So when we have an organism that is diploid, so we have to have, a diploid characteristic. When they're diploid, they have two of each copy. That means you're going to have two alleles per gene. Usually, there's some exceptions. We'll see a little bit later. We'll be kind of keeping it with Mendel easy right now. So for every gene, you're going to have two alleles. So you're going to have two letters, a capital or lowercase or some combination of those. If you have two of the same, right? If you have two of the same, both capital or both lowercase, you are said to be called homozygous. Homo means the same. Zygous is referring to your chromosomes or your genetic information. If you have one of each, right? So a capital and a lowercase, you're called heterozygous. Hetero means different. So the way that we would distinguish these two homozygous is we would call this homozygous dominant, and this is homozygous recessive. Okay, so that's the language you would use when you're describing the characteristics of these organisms. So homozygous dominant would be big letter, homozygous recessive would be little letter, and heterozygous would be one of each, one big letter and one little letter. So depending on whatever letter you assign, and it's arbitrary. Usually people pick the letter of the trait, like plant height. You could use H, or you could use T, or you could use P for purple or whatever. But again, just pick one letter per gene, not a different letter for dominant and recessive. That's a big obstacle. You need to make sure that you are not getting trapped into that. So a couple other terms is when we talk about genotype and phenotype. So the phenotype, this is in reference to the physical appearance, the expression of the gene. What is it looking like outwardly? But it's any expression, because your expressions could be a molecular enzyme or a trait inside of your cells, or it could be a physical feature. And in your labs, in your genetic labs, you're going to be looking at physical features. So for example, tall or short, those are the phenotypes if we're talking about like plant height or even person height. The genotype is the combination of alleles. So those are going to be your letters. So you could be homozygous dominant, you could be heterozygous, you could be homozygous recessive, and then you could describe your phenotype. So when we talk about the phenotype of the genotypes, you can kind of predict which one would be which. So if you have either of these genotypes, big T, so that's our homozygous dominant, or big T, that's our heterozygous, they are going to have the same phenotype, because in this case, they both have at least one allele that is the dominant allele, and dominant alleles overshadow any recessive allele that might be there. So let's say in this particular example, tall is dominant and short is recessive, okay? So if you have either homozygous dominant, which is big T, or heterozygous, which is big T, they will have, both of them will have the tall phenotype. If you happen to have homozygous recessive or little t, you will have the short phenotype, okay? So if knowing this information that tall is dominant and short is recessive, you can start kind of answering some questions. So what if I were to say to you, a pea plant, I would have to give the information. In pea plants, tall is the dominant trait, short is a recessive trait. If you have a short pea plant, what is its genotype? Okay, that's a fair question. So I've given you the information about the plants. Tall is dominant, short is recessive. I've given you the phenotype of the plant, short. You should be able to figure out the genotype based on that information given in the question. So if short is recessive, okay, and it says you have a short pea plant, what is its genotype? Well, for a plant to be recessive and have the recessive phenotype, the only genotype that it could have is little t, little t. If I were to say, okay, in pea plants, tall is dominant and short is recessive, what is the genotype of a tall plant? Okay, so here's the tall plant. Would you be able to 100% tell me what the genotype of that tall plant is? What do you think? The answer is no, because there's two choices. If you have the tall phenotype, you could either be homozygous dominant or you could be heterozygous. You'd need more information to tell specifically whether I was looking for the homozygous dominant or the heterozygous. But you could answer the tall plant could either be homozygous dominant or heterozygous, because both of those answers give you the tall phenotype. So understanding this terminology and this language is super important in being able to solve genetics problems, which the expectation is you should be able to solve genetics problems. All right, the last term is what's called a test cross. So a test cross is, let's say you do have a mystery. I'm going to erase some of my terms here so I can have some space. So let's say in a test cross, you're like, okay, so I know, let's do tall and short. So I'll just write this up here. Tall is dominant and short is recessive. All right, so that's like my key. That's my key of information. So let's say I have a tall plant, and that's all I know. I know its phenotype, it's a tall plant, but I don't know what its genotype is. I want to figure out my tall plant's genotype. I can't tell just by looking at it because it could be big T, or it could be big T, little t. I don't know. It's a mystery. So how can you figure that out? Well, you do what's called a test cross. So a test cross is take your unknown, in this case the tall plant, and cross it with a homozygous recessive. It would have to be a short, right? So if you take a tall plant and you cross it with a short plant, okay, and look at the offspring, that's going to let us know what the outcome is, because we know the short has to be little t, little t. We have to know that because that's the only genotype a short plant can have. If the tall is big T, and we cross those, 100% of the offspring would be hybrids. So 100% of the offspring would be tall. And that would let us know that our original tall plant was homozygous dominant. It's not a mystery anymore. We would have figured it out. But what if it was this one and we had those guys cross? Would 100% of those offspring be tall? No, because there would be a possibility that some of the offspring would be big T, little t, and little t, little t. So you would end up with 50% of this and 50% this. So if you did a cross with your mystery tall with a known short, and half of the plants were tall and half of the plants were short, that would tell you that the genotype of the tall plant had to have been heterozygous. So a test cross allows you to kind of solve some mystery of our original tall plant. Was it homozygous dominant or was it heterozygous? Cross that mystery with a known short, homozygous recessive, and then that will tell you, looking at the offspring, that would tell you what that mystery parent was. All right, so that's a test cross. So here's some vocab. Really make sure you're comfortable. Homozygous dominant, homozygous recessive, heterozygous. Be able to understand what that means. Phenotypes and genotypes. Be comfortable using that terminology. Understand the nature of what a test cross is, knowing that you can solve mysteries by crossing with a known homozygous recessive. If you are having any trouble comprehending, grasping, wrapping your brain around these terms, they get mixed up all the time, drop by a Zoom office hour, send me email questions, because this is really foundational for once we start getting into the application of monohybrid crosses, dihybrid crosses, pedigrees, all the fun genetic stuff that we're going to be getting into in the rest of this chapter. All right, I will see you next time. Bye.